Welcome to the deep dive. We're diving deep today, really deep, going all the way down to the molecular level to explore just how microbes grow.
That's right. We're going beyond just looking at them under a microscope. We're going to get into the nitty-gritty of the machinery inside bacterial cells that actually makes their lives tick.
To do that, we've poured over a ton of research, really tried to bring together a comprehensive view of this complex field.
Exactly. The goal is to give everyone listening a solid understanding of the basic molecular processes that allow microbes to grow, divide, form all those crazy shapes and structures, and even respond to what's going on in their environment.
I think, at least for me, when I think about how microbes grow, it's always like those time lapses you see of bacteria dividing, splitting in two. But obviously, there's a whole lot more going on under the hood. We need to be able to see what's happening at that incredibly tiny scale. It's not like we can just zoom in with a normal microscope. So how has our ability to observe this changed? Like, what are we even looking at?
It's amazing how much our understanding has changed with the advent of new technologies. We used to be stuck with static images, just snapshots frozen in time. But now, we've got things like fluorescent tagging. It's like attaching these tiny little labels, almost like microscopic beacons, to the molecules inside the cell.
So instead of just seeing the overall shape of the cell, we can actually pinpoint specific molecules within it.
Exactly. And one of the most common ways to do this is using something called reporter genes. A prime example is the green fluorescent protein, or GFP, that you might have heard of. Basically, when these genes are switched on, the cell produces a protein that glows, fluoresces, under certain kinds of light. Then with fluorescence microscopy, we can see where that specific molecule is in the cell and how it's behaving.
So it's like shining a spotlight on the parts of the cell we're most interested in. That's cool.
It is, and it gets even better. We now have super resolution microscopy, which can see details at a scale of like 5 to 50 nanometers. That's mind-blowingly small, way beyond what conventional microscopes can do.
So we can practically see the individual molecules doing their thing.
Pretty much. And that allows us to track individual molecules in real time. We can see how they move, interact with each other, and really get a sense of the dynamics of the cell. It's like shifting from still photographs to watching a live video.
So with these incredible new tools, what's one of the most fundamental things we can see when it comes to microbial growth at the molecular level?
Well, for the majority of bacteria, the way they grow is through binary fission. One cell just splits in half, forming two identical daughter cells. It's like the ultimate form of cloning.
And I'm guessing the first step in that process is copying the DNA, making sure each daughter cell gets a complete set of instructions.
You got it. Chromosome replication is absolutely essential, and it's regulated really, really tightly. You don't want any mistakes creeping in when you're duplicating your entire genetic code.
So tell me, how does that process even start, especially in a bacteria like E. coli, which we know so much about?
In E. coli, replication always starts at a specific spot on the chromosome called oriC, the origin of replication. And it's kicked off by this protein called DnaA. But here's the catch. DNA can only do its job when it's bound to ATP. It's like the fuel that activates it, turns it into DNA ATP.
Okay, so you need DNA bound to ATP to start copying the DNA. But like you said, this process is tightly regulated. How does the cell make sure it only happens at the right time?
There's a bunch of mechanisms at play, and they're all fascinating. One of them involves a bit of competition between DnaA and another protein called SeqA. So right after the DNA is replicated, the new strand is only methylated on one side. It's what we call hemimethylated. Now, SeqA has a real strong preference for binding to oriC when it's in this hemimethylated state.
So it's like SeqA is blocking the door, preventing DNA from getting in and starting replication too soon.
Exactly. It's only once the DNA is fully methylated, meaning both strands have those methyl tags, that SeqA lets go, giving DnaA a chance to bind and potentially start a new round of replication. It's a pretty clever way to make sure the cell doesn't jump the gun and start replicating before everything is ready.
I like that analogy, SeqA as the gatekeeper. What are some of the other ways the cell keeps things in check?
Well, the cell can actually control the amount of DnaA that's being produced in the first place. It's like a supply chain control. By repressing the expression of the DnaA gene, it limits how much of this initiator protein is floating around.
So less DNA means a lower chance of triggering replication prematurely. Makes sense.
Right. And then there's this idea of titration. Scattered throughout the chromosome, there are these specific DNA sequences that DNA loves to bind to. They're like decoys. So instead of binding to oriC and kicking off replication, a bunch of DNA gets tied up at these other sites. It's like reducing the concentration of free DNA that can actually do any damage.
And I bet there's a protein involved in taking DNA out of commission, too.
You bet. There's this protein called HEDA, which teams up with a component of the DNA replication machinery called the sliding clamp. Together, they basically deactivate DNA ATP by helping it to break down its ATP. So the active DNA ATP turns into inactive DNA ATP, which can't start replication.
So it's like a multi-pronged approach, controlling the production, availability, and even the activity of DNA. The cell is really serious about making sure replication happens at the right time.
Oh, absolutely.
Wow.
It's all about precision and timing. And, when bacteria are growing really fast, they need to speed things up even further.
How do they do that? Do they somehow make replication go faster?
Even better. Imagine this, they can have multiple replication forks running at the same time on a single chromosome. It's like having multiple construction crews working on different sections of the same building.
So instead of waiting for one round of replication to finish, they start a new one before the first is even done. Talk about efficiency.
Exactly. It allows them to churn out copies of their DNA much faster, which is essential when they need to divide rapidly to take advantage of good conditions. Now, once you've got 2 copies of the DNA, you've got another challenge. How do you make sure each daughter cell gets one?
That's chromosome segregation, right? Making sure the genetic material is divided equally.
Exactly. And in E. coli, the way they do this is still not fully understood, but it seems like the process of replication itself plays a big role. As the new DNA strands are synthesized, they take up more and more space, almost like they're pushing each other apart.
So it's almost like they naturally get shunted to opposite ends of the cell just by the sheer bulk of the new DNA.
That's a simplified way to think about it, but it seems to be part of the story for E. coli. Other bacteria, though, have developed more sophisticated mechanisms. Take Colobacter again. They've got this partitioning system called the PAR system, which involves a bunch of proteins like para, PAR-B, and POP-C.
Sounds complex. What do those proteins do?
PAR-B is like a scout. It binds to specific DNA sequences near the origin of replication. PAR-A is the mover. It forms these filamentous structures that can actually pull things around. And then there's POP-Z, which hangs out at the poles of the cell, acting as a kind of anchor point. Through their interactions, they make sure the chromosomes end up at opposite ends of the cell, ready for division.
So it's a more deliberate, active process of separating the chromosomes. And I imagine plasmids, those smaller pieces of DNA, also need a way to get distributed to the daughter cells.
Plasmids have their own segregation mechanisms too, often involving similar systems to the chromosomes. It makes sure that each daughter cell inherits at least one copy of every plasmid so those extra genes don't get lost.
Okay, so the genetic material is all copied and sorted. Now the cell needs to physically split in 2.
Yeah.
You mentioned this protein complex called the divisome. Tell me more about that and the role of these FFTs proteins.
The divisome is like the construction crew that actually builds the wall between the 2 daughter cells. It assembles right at the spot where the cell is going to divide and coordinates the whole process. And one of the key players in this crew is a protein called FTKS.
FTSZ, that sounds familiar.
It's actually related to tubulin, the protein that forms those microtubules in our own cells, which are part of the cytoskeleton. It's amazing to think that a bacterial division protein is structurally similar to a key part of our cell's internal scaffolding.
Wow, so it's a bacterial version of a protein we also have. What exactly does FTSZ do during cell division?
It's the one that forms the actual division ring. FTSZ molecules come together and form this ring-like structure at the exact center of the cell, basically marking the spot where the new wall will form. And the location of the ring has to be really precise, especially in rod-shaped bacteria like E. coli. You want that division to happen right down the middle.
How do they make sure the ring forms in the right place? It can't just be random.
It's not. And a lot of that precision comes down to a group of proteins called the MIN proteins. Min, Sci, Mind, and Mine. They work together in this fascinating way. Min, Sci, and Mind are like bouncers. They prevent FTSE from forming its ring anywhere it wants. They actually oscillate back and forth along the cell, spending most of their time near the poles and less time in the middle.
So they keep FTSE away from the ends of the cell.
Right. And then you've got Mine, which is like the party planner. It hangs out in the middle of the cell and helps to clear out Min, Sci, and Mind, making space for FTSE to assemble its ring. So you end up with this gradient where the concentration of Min, Sci, and Mind is lowest in the middle of the cell. That's where FTSE can finally come in and build the division ring.
I like those analogies. It definitely helps to visualize it. So the Min proteins are like a patrol, making sure the ring forms right where it's supposed to.
Exactly. They're essential for making sure cell division happens in a controlled way. And it's not just E. coli. In Colobacter, a protein called MIPC plays a similar role, but it uses a different mechanism, interacting with the chromosome itself to influence where the FTSZ ring forms.
So different bacteria can have slightly different ways of controlling the same process. And besides FTSC and the Min proteins, who else is involved in this divisome complex?
Oh, there are a bunch of other FTSS proteins, each with their own specific job. IfKSA and ZipA, for example, are like the anchors. They attach the FTSC ring to the cell membrane, making sure it's firmly in place. And then there's IfTSI, which you might remember from our talk about peptidoglycan synthesis.
Right, if TSI is one of those penicillin-binding proteins involved in building the cell wall.
Exactly. And it highlights why certain antibiotics, like penicillin, are so effective. They interfere with the activity of PDPs, which are essential for cell wall synthesis, and therefore for cell division as well. There are other FTs proteins that help to coordinate the constriction of the cell envelope, making sure everything happens in sync, and once the division ring has done its job, it needs to be taken apart. So FT-ESAC actually disassembles, and this triggers the ingrowth of the septum, the new wall material that separates the two daughter cells.
Okay, so we've covered how bacteria divide, but what about their shape? Some are rods, some are spheres, some are even curved. What determines these shapes? It can't just be random, right?
Definitely not. Bacterial shapes are incredibly diverse, and a lot of it comes down to their cytoskeleton, even though it's not as elaborate as the cytoskeleton in our own cells.
So bacteria have a cytoskeleton too. That's interesting.
They do, and it plays a big role in determining their shape. One of the key proteins is mevribi, which is related to actin, a major component of the eukaryotic cytoskeleton.
Actin, that's the protein that helps give our cells their shape and allows them to move. So what does microbial do in bacteria?
In rod-shaped bacteria, mirabe forms these long filaments that run along the length of the cell just beneath the membrane. Think of it like the scaffolding that supports the cell's shape. These filaments also act as tracks for the elongisome, the machinery that synthesizes peptidoglycan, the main component of the cell wall.
So microbe guides the construction of the cell wall, ensuring the cell elongates into that rod shape. What happens if you mess with microbe?
You get shape-shifting bacteria. If you disrupt mirabe, either with drugs or by messing with its genes, rod-shaped bacteria will often lose their elongated shape and become spherical, like little kochi. It's a pretty dramatic demonstration of how important mirabe is for maintaining the rod shape.
So mirabe is like the master architect of the rod-shaped cell. What about curved bacteria, like Caulobacter? What gives them their unique shape?
Caulobacter uses another protein called crescentin, which is actually related to keratin, a protein we find in our hair and nails. It's crazy to think that a bacterial protein is similar to something that makes up our hair.
So Caulobacter has a bit of a hair protein that helps it curve. How does that work?
Crescentin localizes to the inner concave side of the curved cell. It seems like the polymerization of crescentin filaments on that side creates a force that bends the cell into that crescent shape. So different cytoskeletal proteins contribute to different shapes. It's like the bacteria have their own internal toolkit for shaping themselves. And you mentioned earlier that alpha proteobacteria, the group that Caulobacter belongs to, are quite diverse in terms of their shapes and growth patterns.
Yes, it seems like they're really good at adapting to different niches.
Absolutely. Agrobacterium tumefaciens, for example, grows by elongating at one pole of the cell. It's like it's always pushing forward in one direction. And then there are bacteria that reproduce by budding, where a little outgrowth forms on the mother cell and eventually pinches off to become a new daughter cell. It's a completely different way of growing compared to binary fission.
So many different strategies for multiplying. Okay, let's get back to the cell wall. We've talked about peptidoglycan a bit, but why is it so essential for bacteria?
Peptidoglycan is like the armor that protects bacteria. It forms this mesh-like layer around the cell membrane, giving it rigidity and protecting it from the high internal pressure. Without it, most bacteria would just burst open.
So it's essential for maintaining their shape and keeping them from exploding. How is this important structure actually made?
It's a multi-step process that involves both the cytoplasm, the inside of the cell, and the periplasm, the space between the inner and outer membranes in gram-negative bacteria. First, you've got the precursor molecules, which are these sugar units linked to a short peptide chain. These are synthesized inside the cell.
What happens next? How do they get outside the cell to build the wall?
They need a special transporter, and that's where bacterial comes in. It's a lipid molecule embedded in the membrane, and it acts like a ferry, carrying these peptidoglycan precursors across the membrane to the outside.
So bacterial shuttles the building blocks of the wall to where they're needed. What happens once they're outside?
First, you need to make space for the new material. So enzymes called autolysins come in and carefully break down small sections of the existing peptidoglycan network. Then other enzymes, called transglycosylases, insert those new precursors into the gaps, linking them together.
And then comes the cross-linking, right? That's where those penicillin binding proteins we talked about earlier come in.
Exactly. PBPs catalyze the formation of peptide crosslinks between the glycan strands, making the peptidoglycan layer strong and rigid. And remember, this is exactly what penicillin and other beta-lactam antibiotics target. They bind to PBPs and prevent them from doing their job, which weakens the cell wall and eventually leads to bacterial death.
So by understanding how peptidoglycan is made, we can understand how a whole class of antibiotics works. That's pretty cool.
It really highlights the importance of understanding these fundamental molecular processes. And it's not as simple as you might think. Some PBPs can actually do multiple jobs. They have both transglycosylase and transpeptidase activity. And there's another family of enzymes called SEDS proteins that also contribute to building the glycan strands of peptidoglycan. So it's a complex process with multiple pathways.
Bacteria always seem to have a backup plan, don't they? Okay, so far, we've focused on individual bacteria growing and dividing. But microbes are also known for their ability to differentiate, to form these complex structures and communities. Let's talk about the regulation of development, starting with endospore formation in bacillus.
Endospore formation is a fascinating survival strategy that bacillus uses when times get tough, like when nutrients are scarce. It's basically like going into hibernation. The bacterium divides asymmetrically, creating this incredibly resistant endospore inside a larger mother cell.
So it's like building a bunker to wait out the bad times. And these endospores are known for being super tough.
They are incredibly resistant to heat, radiation, chemicals, you name it. They can survive for incredibly long periods of time until conditions improve. And the whole process is triggered by a complex signaling pathway that ultimately activates a protein called SeqA. The SeqA is like the master switch, flipping the cell from growth mode to spore formation mode.
Once that switch is flipped, what happens?
It sets off a cascade of events where different genes are activated in a specific order. It involves the sequential activation of alternative sigma factors, which are proteins that control which genes are transcribed. Different sigma factors become active in the forespore, the part that will become the endospore, and in the mother cell, ensuring that the right genes are expressed at the right time and in the right place.
It's like a carefully choreographed molecular dance, with each step leading to the next.
Exactly. And to make things even more complex, there are also anti-sigma factors, which can bind to sigma factors and block their activity. So the timing of sporulation also involves overcoming these inhibitors at the right moment.
And to add to the drama, you mentioned that some bacillus cells can even become cannibals during sporulation.
It's true. Under extreme nutrient limitations, some sporulating cells will actually kill and consume their non-sporulating siblings, using those nutrients to fuel their own spore formation. It's a pretty brutal way to ensure survival.
Talk about survival of the fittest. Once those endospores are formed and they've weathered the storm, how do they come back to life when conditions become favorable again?
Germination is a three-stage process that begins when the spore senses that conditions are right for growth again. The first stage is activation, which is often triggered by nutrients binding to specific receptors on the spore's surface.
So they're essentially sensing food in the environment.
Right, and that sets off a chain reaction. The second stage is germination itself, where the thick protective layers surrounding the spore are broken down, and water rushes in, rehydrating the spore. And finally, there's outgrowth, where the spore becomes metabolically active again, starts making new proteins and DNA, and eventually divides to form a vegetative cell.
From dormant bunker to active cell, it's a pretty remarkable transformation. Let's shift gears now and talk about another model organism for studying development, Caulobacter, which has a very distinct life cycle.
Caulobacter is fascinating because it has these two very different cell types, a motile swarmer cell and a sessile stalk cell. The swarmer cell has a flagellum, a tail that lets it swim around and explore, but it can't replicate its DNA or divide.
So it's the explorer, the adventurer.
Right. And then after a while, it settles down and turns into a stalked cell. The stalked cell attaches to a surface and grows a stalk at one end, which acts like an anchor. This stalked cell is the one that's specialized for DNA replication and cell division. It divides to produce a new swarmer cell, which swims off to explore new territory, and another stalked cell, which stays put and keeps replicating.
So they alternate between exploration and reproduction. How is this cycle controlled at the molecular level?
It all comes down to the precise regulation of three key proteins: ChRA, EcRA, and DnaA. These are transcription factors, meaning they control the expression of other genes. Each of these proteins is active at a specific stage of the cell cycle, ensuring that the right processes happen at the right time. ChRA, for example, is active in the swarmer cell and prevents DNA replication, while DA is active in the stalk cell and promotes replication. So it's like a carefully timed relay race, with each protein handing off the baton to the next.
A well-coordinated molecular dance. It's amazing how similar this is to the regulation of the cell cycle in our own cells, even though bacteria are so much simpler.
It really highlights the fundamental nature of these regulatory mechanisms. Okay, let's talk about a different kind of developmental process that happens in filamentous cyanobacteria heterocyst formation in Animana.
I remember learning about heterocysts. They're those specialized cells that fix nitrogen, right?
That's right. Nitrogen fixation is a crucial process for life, but the enzyme that does it, nitrogenase, is incredibly sensitive to oxygen. It gets destroyed by oxygen. And the problem is, Animana are photosynthetic, meaning they produce oxygen as a byproduct.
So how do they solve that problem? They can't just stop photosynthesizing.
They don't. Instead, they've evolved this amazing solution where, when nitrogen is scarce, some of their cells differentiate into these specialized heterocysts. These heterocysts are basically anaerobic chambers. They create an oxygen-free environment where nitrogenase can function.
So it's a division of labor. Some cells do photosynthesis and some fix nitrogen in these protected compartments. How do they decide which cells become heterocysts?
It's triggered by low nitrogen levels in the environment. When nitrogen is scarce, a molecule called alpha-ketoglutarate builds up inside the cells, and this signals the activation of a protein called NTCA. NTCA is a transcription factor, and it activates the expression of another gene called HdaR.
And what does HdaR do?
HdaR is the master regulator of heterocyst development. It controls the expression of a whole bunch of genes that transform a regular vegetative cell into a heterocyst. And one of the key changes is that heterocysts shut down photosystem 2, the part of the photosynthetic machinery that produces oxygen.
That makes perfect sense. They're essentially sacrificing their ability to produce oxygen to create an environment where nitrogen fixation can happen?
Right, it's a really clever adaptation. And there's also a fascinating mechanism for ensuring that the heterocysts are spaced out evenly along the filament.
I remember reading about that. It's not like they just form randomly.
It's all about communication. Developing heterocysts produce a small peptide called PATTES, which diffuses to neighboring cells and inhibits their differentiation into heterocysts. So you end up with this pattern where heterocysts are spaced out at regular intervals, ensuring efficient sharing of resources and division of labor along the filament.
It's incredible how bacteria can communicate and coordinate their behavior like that. Okay, let's move on to our last topic on development biofilms. We hear a lot about biofilms, but what exactly are they and how do they form?
Think of a biofilm like a microbial city. It's a community of bacteria that have attached to a surface and encased themselves in this slimy matrix, which we call EPS, or extracellular polymeric substance. It's a mixture of polysaccharides, DNA, proteins, all sorts of stuff.
So it's like they're building their own little ecosystem, a protective layer around themselves. How does this process even start?
It usually begins with individual bacteria, planktonic cells, swimming around and encountering a surface. They attach to the surface, and if conditions are right, they start to multiply and produce this EPS, forming a microcolony. As the colony grows, they communicate with each other, and the biofilm develops into a more complex three-dimensional structure with channels and different layers.
And I know quorum sensing, that bacterial communication system, is important for biofilm formation, right?
Absolutely. Quorum sensing allows bacteria to sense how many of their buddies are around. As the cell density increases, they release signaling molecules, and when those molecules reach a certain concentration, they trigger changes in gene expression. This can lead to the production of more EPS, changes in the biofilm architecture, or even the dispersal of cells from the biofilm to colonize new areas.
It's like they're voting on what to do next as a community. And you mentioned earlier that in Pseudomonas aeruginosa biofilms, some cells actually sacrifice themselves to contribute to the biofilm matrix.
It's true. A subpopulation of cells undergoes programmed cell death, and the DNA and other molecules they release actually become part of the structural scaffold of the biofilm. It's like they're giving their lives for the good of the community.
That's dedication. And is biofilm formation always triggered by high cell density?
Not necessarily. Different bacteria have different strategies. In Vibrio cholerae, for example, low cell density actually promotes biofilm formation, while high density leads to dispersal. So it really depends on the species and the environment. And biofilms aren't just a bacterial thing. Some archaea, like Sulfolobus acidicaldarius, also form biofilms, showing that it's a widespread strategy in the microbial world.
Okay, we've covered a lot of ground on how microbes grow, divide, differentiate, and even form communities. Let's shift our focus now to how we try to control them with antibiotics. How do antibiotics actually work at the molecular level?
Antibiotics target essential processes in bacteria, things they need to survive. Ideally, they target pathways or molecules that are different in our own cells so they don't harm us.
So they're exploiting the differences between bacterial and human cells. Can you give me some examples of these targets and the antibiotics that attack them?
Sure, DNA replication is a major target, and quinolones are a class of antibiotics that interfere with this process. Other antibiotics, like rifampin, target RNA synthesis, the process of making RNA from DNA.
What about protein synthesis? That seems like a pretty essential process.
It is, and there are many antibiotics that target different steps in protein synthesis, like tetracycline and streptomycin. And of course, we've already talked about the beta-lactams, which target cell wall synthesis. Some antibiotics, like daptomycin, go after the cell membrane itself.
So we have a whole arsenal of drugs that attack different parts of the bacterial cell, but bacteria are notorious for developing resistance to antibiotics. How do they do that?
They have a number of tricks up their sleeves. One way is by changing the target itself. If the antibiotic can no longer bind to its target because of a mutation of the bacterial gene, the drug becomes useless.
It's like changing the locks on your house so the key no longer fits.
Exactly. And another way is by producing enzymes that destroy the antibiotic. Beta-lactamases, for example, break down the beta-lactam ring that's essential for the activity of penicillin and its relatives. It's like having a molecular shredder that destroys the drug before it can do any harm.
What other defenses do they have?
Some bacteria have these efflux pumps, which are like tiny pumps that actively pump the antibiotic out of the cell. It's like having a security system that throws out any intruders.
So they prevent the antibiotic from building up to a high enough concentration to be effective.
Exactly. And some bacteria have even developed metabolic bypasses. They find a way to do the essential job that the antibiotic is trying to block, but using a different pathway or enzyme that's not affected by the drug. For example, MRSA, methicillin-resistant Staphylococcus aureus, has a modified penicillin-binding protein that methicillin can't bind to, so it can still build its cell wall even in the presence of the drug.
So they find a workaround, a detour. Where do these resistance genes come from in the first place?
They can arise through random mutations, but the bigger problem is horizontal gene transfer. Bacteria can share genes with each other, even between different species, and this includes resistance genes. So once one bacterium develops resistance, it can quickly spread to others.
That's a scary thought. So it's not just about individual bacteria developing resistance, it's about the spread of resistance genes through the entire bacterial community.
Exactly. It's a constant arms race.
Right.
And then there's the phenomenon of persistence, which is a bit different from resistance.
What's the difference?
Persistence isn't about genetic changes. It's about a small subpopulation of bacteria entering a dormant state where they're not actively growing. Many antibiotics work by targeting processes involved in growth, so these dormant cells are essentially invisible to the drug.
So they're hiding out, waiting for the danger to pass.
Exactly. And once the antibiotic is gone, they can wake up and start growing again. This dormancy is often associated with toxin-antitoxin systems, where a toxin inhibits cell growth, but an antitoxin keeps it in check. Under stress, the antitoxin can be degraded, unleashing the toxin and forcing the cell into dormancy.
So stress can trigger this temporary shutdown.
Right. And another contributor to dormancy is the stringent response, which is a global regulatory mechanism that's activated when bacteria are starved for nutrients. It leads to a shutdown of growth and a shift towards survival mode. And the interesting thing about persistence is that it's not all cells that become dormant, only a small fraction. This shows that even within a population of genetically identical bacteria, there can be significant variation in how they respond to stress.
It's like they're hedging their bets, ensuring that at least some of them will survive. Wow, this has been an incredible journey into the molecular world of microbial growth. We've gone from the basics of cell division and shape to the complexities of differentiation, community formation, and even the strategies bacteria use to survive our attempts to control them with antibiotics.
It's amazing how much is happening in these tiny organisms, and we're learning more and more every day thanks to advances in microscopy and other techniques.
And the more we learn, the more we realize how adaptable and resilient microbes are. It really makes you wonder what the future holds for a fight against antibiotic resistance and how we might be able to harness the power of microbial development for good. Thanks for joining us on this deep dive, and we'll see you next time.